Okay, Paul, we are recording. So welcome everybody to our first PacBio Lunch and Learn session. I am your co-host, Sarah Fardy from Millennium Science. I'm the marketing manager here, and we have with us Paul Gooding. So Paul, I'm going to take it away with you. Go ahead.
Okay, great. Thanks, Sarah. And welcome everyone. This is the first of a four-part Lunch and Learn series, just talking through some of the technology and applications with PacBio sequencing on the Sequel II system. So today is hopefully quite a general overview of how the technology works and its benefits and its performance and such like. So we'll take you through really what you can see here and see how we go. So yeah, we'll talk about how the technology works, as I say, and then how well it performs, and then we'll move briefly onto the products and the workflows, and then the applications. And the applications we'll skip through today is just a general overview. We'll concentrate a little bit more on those applications over the three following sessions. They're a fortnight apart. And then we might just show just a couple of the general resources that you can find on the PacBio website if you want more information, or you can reach out to myself or anybody at Millennium Science who distributes PacBio in Australia and New Zealand. So let's talk about the technology. There's some nice little graphics here. So going back a slide here. On the right, you've got the Sequel II system itself, just with the drawer open there for the deck. And on the left, you've got a picture of a smart cell, which is where all the magic happens. This is where the sequencing is actually done on this smart cell. And the little circle in the middle is a kind of interpretation of what that smart cell is actually like inside in detail. So as I say, we'll go here now. So on the left, what you've got in that smart cell is a metal coated glass sort of slide or chip, and there's tiny little nano fabricated holes, nano holes in that metal coating that goes up to the glass. And actually on top of the glass there's some biotin that's also there that we'll come to later. And on a smart cell ATM on the Sequel II, you've got about 8 million of these little nano holes in. They're called ZMWs, or zero mode waveguides, and that's the little vesicles, little reaction vessels where the sequencing takes place. And if we go across to go to a little video there as it clicked in, each of these ZMWs are illuminated from the bottom, so you're able to shine light through the glass substrate and into the bottom of the wells. But a little bit like your home microwave, the size of the part of the hole here compared to the wavelength of the light only allows the light to penetrate a very, very, very small distance. The door of your microwave oven at home has got a mesh over it, you may have noticed. And the reason that keeps you safe is because the wavelength of the microwaves can't get through the holes in the mesh, but it's enough to be able to see your food cooking inside. So that's how it works. And it's a similar principle here. The light only just gets through the bottom. And this is important. So what happens with your sequencing? This is single molecule sequencing that we do here on a PacBio. So you've got your template DNA that you want to sequence, which is here in purple. And you anneal a sequencing primer, which you can see in yellow, and bind a polymerase. Now the polymerase has steptavidin bound to it, so it sticks to the biotin at the very bottom of the wells. But what happens, the sequencing, the addition of each base happens at the very, very bottom of the well. This allows you to put on the fluorescent nucleotides, which allows you to do your sequencing in the way that a lot of other sequencing systems work. You have a fluorescent molecule for the G's, T's, A's, and C's, different color for each one. But those fluorescent molecules that are floating on the nucleotides don't get into the light source, so they don't get seen. So the background is very, very low indeed. Only the base that's about to be incorporated into the DNA by the polymerase as part of the sequencing, do you get the fluorescent signal. So that's how it's measured. So each base is added, that fluorescent nucleotide is held by the polymerase very, very briefly, and then you get the image of that light, and it's a very stable process. It's happening in real time, and the time gap is very precise. And we'll talk about why that can be important too, very briefly too. So here, as I said, you've got your G's, A's, T's, and C's. Each one has a different colored fluorophore. As it becomes incorporated into the molecule, as the polymerase acting as that sequencing engine, if you like, it incorporates the base, and it actually slices off the fluorescent molecule, the dye, as it's added. So you end up with nascent DNA, which is perfectly normal DNA, no fluorescent molecules on it. Hence, it allows it to sequence very, very long, because you don't get background build-up of any fluorescent molecules. And again, on the figure C there, kind of another representation of how a fluorescent base is going to be added into the chain, and just as it's added, it's held. We can run that as a little video, I think. See if it wants to run for me. No, doesn't want to run for some reason. Oh well, you get the idea. You know how DNA works. Each base is added, and then it's a different color fluorophore, and the cameras that run a movie over this whole chip are able to see the different colors in real time and build the sequencing reaction. So again, this is the sort of step-by-step. You can see here, hopefully it's big enough on your screen. There's an A about to come into the molecule. As it's come in and held at the bottom and is illuminated by the light source, you get a flash of light. So bang, you get signal. The background signal is very, very low, because I said the other fluorescent molecules are floating in solution away from the light source that illuminates them. So you've got very, very low background, then bang, you get signal, you get an A incorporated here. Once it's incorporated and the fluorophore is sliced off and the polymerase moves on, you've got very, very low background. You get your next base coming in here at T, bang, you get signal, and so on and so forth. And this chemistry is very precise in timing. So you're seeing this in real time on the video. And there's other things that you can elude from this data, which is we will talk about briefly today. But bear this in mind, that you're getting effectively a piece of signal, then blank, and a piece of signal. And this is in real time. So you've actually got the known distance in time between these additions of bases. And that's a very precise piece of chemistry, depending on the buffing solutions and everything that you have here in the polymerase. So here, the deck on the left is open. You've got your smart cells loaded onto the deck. You can load 8 smart cells onto the Sequel 2 here. So you can run 8 of these experiments in one run. So it's a sort of walk away time of about 3 or 4 days to run it if you're running it on the full movie times. And there was a little video that played in the middle here of the detection that we said. So you're incorporating a base, you get a flash of light, then there's background almost zero, and then another flash, and so on. And it's a very precise timing for each base addition. And on the right-hand side here is essentially a snapshot from the movie that's being taken. It's a continuous movie that's taken of the smart cell. And each flash of light is recorded, and then very smart software is able to interpret the flash of light in order from each spot and turn that into base calls. And the movie times can run up to 30 hours, so you can get very, very, very long read sequencing. And there's that movie running again. Can we get off that? Okay, good. Okay, so some of the advantages, I guess, of smart cell sequencing with PacBio. Why would you use it? There are other sequencing facilities and machines available. Well, just starting with these little 5 points here, it is single molecule sequencing. There's no PCR amplification going on for your molecules. You're not having to build a cluster on a slide like you do with some other systems. It's purely single molecule. It's not an amplified piece of DNA like Sanger sequencing. Single molecule, so you're sequencing absolutely what you get from each molecule. So if you have variation in those pieces of DNA in your sample, say if you were sequencing a transcriptome and you wanted to look for alternative splicing or something like that, you're seeing all the differences from all those single molecules. So that's important. It's also very, very accurate. In fact, arguably probably the most accurate sequencing you can actually do out there now, up to Q50 scores. And we'll talk about how that's done as we go through. And as I said, it's long reads. very long reads. You can read up to sort of 300 kb for some of those molecules, but certainly 20 kb fragments can be sequenced at very, very high accuracy. Very uniform coverage. What does that mean? Well, it's not particularly biased with homopolymer regions or GC content, high GC, low GC. It's all the same. It's not prone to errors in those sorts of things. So it's very, very uniform coverage, and any errors that occur in the sequencing are totally random. And you'll see from how the sequencing works why those can be screened out, and you can get this incredibly high accuracy consensus sequencing. Oh, and finally, just the epigenetics there. I will just point at that and say you can also measure base modifications due to that timing that we saw earlier between the base additions. When there's a methylated base, it alters the timing, and the software can see that in real time and call that there must have been a modified base like a methylated A or something in a GATC repeat. So what does it do? Well, as we said, long reads, tens of kilobases, in fact, a lot longer than that. It can sequence comfortably 15, 18, 20 kb library fragments, anything from 500 up to 50,000 base inserts, and with very long reads and very, very high accuracy. As I said, Q50 scores, that's 99.999% accuracy in some cases, which is fantastic. And the long reads, you're able to screen anything out below Q20, and that's 99% accuracy. So all the data you get is very high accuracy. As I said, it's not worried too much by GC content or homopolymers or anything that's particularly strange, like repeats and things in the sequence. It's able to sequence through it, and you're also able to. What's going on here? Sorry about that. You're also able to see some epigenetic modifications in the sequence due to the timing. There's not a separate sample prep need to do where you have to do these special modifications to read the methylated bases. So again, little video running on the right, and we will concentrate a lot more on what's going on really on the right-hand side here, that just because of the way the sequencing works, it effectively sequences in two modes. So you don't exactly set things up for this, but it sequences either as a continuous long read, or more commonly now set up to do this, what's called circular consensus sequencing mode, or CCS, or HiFi to its friends, because it's the high fidelity reads, very, very highly accurate. With the CLR, the continuous long read, you can put your DNA molecules in, and you make these little smart bells, as they're called. So you ligate adapters on the end to make the molecules a circular molecule. Your sequencing primer and polymerase binds on the smart bell end, and you have a very, very long piece of DNA, and it sequences and sequences away. And basically that continues to run until either the sequencing runs out of steam or your movie time is up. And you can set your movie time, as I said, up to 30 hours, but for some cases you might want to set it only for 10 hours or 20 hours or whatever. And so you get a very, very long read, and that's great. But it was noted as the system performance has improved, and people were size selecting for their library fragments, so everything was very, very even, that what would tend to happen if you had, say, a 15 kb molecule, but let's say you get a piece of sequencing that's 150 kb, what's happened? It's gone around the same molecule 10 times, right? And that's sort of represented on the right here. So it starts sequencing, and it sequences the yellow strand. It goes around the little smart bell adapter on the end, and then it sequences back along the reverse strand in the purple, and so forth, yellow, purple, yellow, purple. So you sequence the same molecule multiple times, and that's how it's able to get such high accuracy. If there happens by chance to be a random error, say here, but it doesn't occur anywhere else, by consensus, you can screen out that error, and over the course of all these bases in all the different molecules, just random errors that occur anywhere get screened out by the consensus mode. So hence the very high fidelity, HiFi, highly accurate sequencing. A few graphs, don't be put off. I know it's your lunchtime, but we'll just talk through these briefly. So here's some data from, it was a 20 kb human library that was run on a Sequel II, and it went into this HiFi mode sequencing, and this shows the read length distributions and base yield performance. So the library was sheared and size selected for 20 kb inserts. And you see here the number of raw bases was over 350 gig. Got just over 4 million total reads off the smart cell, and the longest reads were over 300 kb. What's interesting here is that over half the bases that are in reads that are over 190 kb in length. So very long read. And obviously this helps with mapping and all of those sorts of things. So looking at the data in a slightly different way here. So you're looking at the accuracy. The light blue on the left-hand side is any reads that had a Q-score below Q20. So they're automatically screened out by the analysis software, which is called SmartLink, and we'll talk very briefly about that at the end. But as you can see, the read score quality, there's reads right up there at Q50 score, but even the average is up around sort of just under Q40, so Q38 or something like this, which is very, very high. And in this particular example, again from a 20 kb human library, you're talking about the mean HiFi read accuracy of 99.92%. So very, very highly accurate sequencing. It's a slightly different example, just looking at different size pieces of DNA that are being sequenced here, and the movie times that you can reduce, obviously, as you're running shorter sized library fragments. You don't need to sequence for 30 hours if you've only got a 1.5 kb fragment, because you don't want to sequence it through hundreds of times. A few times through is quite enough to give you this HiFi sequencing accuracy. And as you can see, the Q20 and Q30 scores on the table there, and how many reads you're getting from each. Just to briefly touch on the continuous long read, this is becoming less frequent, and more of the applications, I guess, for PacBio moving towards the HiFi, the CCS type reads, but the continuous long read is still used for certain applications. And again, you can see some of the metrics from here. Half the bases are over 50 kb in this example, and you're still getting reads up over 175 kb, and the accuracy is still good from these types of reads. Just to talk about how these the consensus, the high accuracy of the HiFi or CCS reads works. This is a graph that shows you the quality of your sequence against the coverage. And as we were saying, sequencing a random molecule gives you this consensus accuracy to screen out any errors that occur by random. And as you can see, a coverage of about 30-fold on the graph hits you that Q50 score. So I mean, even at 20-fold, you've got about 45 Q45 scores. So very, very accurate indeed. And an example of how coverage is very even regardless of GC content. So even at 10% GC right up to 80-odd% GC, the coverage is very, very even and very, very good across all sorts of sequence there. And this is the epigenetics that's happening. If you look in the tables here, particular top right here is a good thing to look at, because we said about the time of incorporation of each base is very, very even. Well, if you look at these time scans and you suddenly see a gap occurs here, so the A here in a GATC little motif here where you've got a methylated A. This happens in prokaryotic DNA. The A is bound by the complex, and therefore you get the flash of light from the fluorescent A that's there. but the polymerase stalls very slightly in order to cleave it and bind it onto the extension of the DNA chain. So you get this slight delay in timing before it gets to the T. And of course, if you're doing this in HiFi type reads, you're going to then see it on the opposite strand as well, because it's a palindrome. So you've got the GATC, and this is represented by the little chart here. So you see this delay in time, and the software is able to read this as like there's something weird going on. There's a slight stall on the polymerase. I think looking at the sequence, that had to have been a methylated A. So hence you're calling epigenetic modifications as well within the sequencing. So moving on a little bit to the workflow and what's available. There is a library prep kit. The library template prep kit 2 is the latest version. Very, very easy to use. It's really a pipetting exercise into a single tube. You've shared your DNA if you're running from genomic DNA, and the template prep kit takes care of tidying up that DNA and getting into a state to make it into these smart bell libraries. So it's the shared DNA will have very uneven ends depending where it's shared. So it's able to tidy up those ends and polish them up so then it can tail, and then you can ligate on these smart bell adapters, which are T-tailed, and you can get barcoded adapters as well if you're multiplexing. We'll touch on that in a moment too. The library prep time is comfortable in a day, half a day if you're really pushing, and to get it done, it's a 4 to 6 hour protocol to get the libraries made, and accepts a wide variety of sample types and obviously insert sizes depending how you've shared your DNA, or whether you're working with amplicons or whether you're working with cDNA from your transcripts, from your RNA transcripts. A lot of groups now size select to get a very tight distribution of the sizes they're sequencing. It makes the sequencing work much more efficiently, and as I said, there's multiplexing barcode solutions available. The sequencing itself, you're sequencing on up to 8 smart cells. There was a picture of that earlier, of the 8 smart cells loaded in their trays on the deck of the Sequel II. You've got movie acquisition time up to 30 hours, as little as 30 minutes for some applications, and obviously for shorter molecules you don't need to run for very long. And you can set the machine up. You can see at the bottom there with the 4 trays in the machine, you can set up and have all the reagents all on board with your samples, and walk away over a long weekend like the one that's just coming up, and have your 8 smart cells sequenced. And even data analysis kicked off through SmartLink, and we'll talk about that. There's lots of analysis pipelines built into the system that allows you to do de novo assembly, for example, or variant detection if you're looking at indels or deletions and SNPs and those.
It seems we have lost Paul, perhaps. If anybody can still hear, please comment in the chat box. We'll see if we can get Paul back. Oh, Paul, here we are. We're back.
If you can hear me, we've just had a power cut.
Oh, no.
So,
okay, well, we've got you here. Have you got enough battery to continue?
I think I have, yes, but that was entertaining for everyone. It's really real, this.
I was eating lunch, so that was entertaining for me too. I am here. Thank you.
Unfortunately, I'm running from a laptop. It's just that it was just the internet that I've completely lost, but I'm now hotspotting from my phone. So I'm having work done on our house, and my daughter's just informed me that one of the guys outside may have hit a cable. So that's going to be fun to sort out, but not anybody's problem here other than mine after this lecture. So we'll sort that out in a while. What was I saying? Data analysis. Yes, SmartLink on the server system that comes with the SQL 2 will do a lot of analyses for you, such as de novo assembly and transcript alignment and mapping from ISO-seq type experiments too. So how does it all work here? Let's have a look. This is the template preparation. As I said, you've got your DNA sample. You fragment that with shearing, so Cavaris or Megaruptors or however you want to do your shearing, often mechanical type shearing. That DNA is very uneven. As I said, it's got all the fragmented ends. So you polish those ends up, you damage repair and end repair. It's really important to do damage repair as well, not just the ends that are messy. If you've got any nicks in the sequence, this can cause the polymerase to stall or fall off the molecule, and so you stop getting these nice, really lovely long reads. So it's important to do really good damage repair to your DNA. The analogy is for your polymerase is a bit like your car driving along a nicely made freeway compared to driving off road on a bumpy dirt track. And yeah, the polymerase will fly along the smooth road when your DNA is in good shape, but it's going to shake it to bits driving along the potholed track. So you want your DNA to be nicely repaired and also tidied at the ends on the end repair, so you can ligate the adapters. Say there's a tailing step on here, so they're T-tailed adapters now to go onto an A-tailed molecule to make the ligation a little bit better. Lots of size inserts available, and the amount of DNA is just that you require to set up the experiment is just in the chart at the top there. So if you're going for small insert sizes like amplicons, then you just need 200 to 500 nanograms, that kind of amount of sample. If you're going for size selected large high molecular weight DNA, then you need maybe 3 to 10 micrograms, that kind of thing for shearing, and because there's a lot of cleanup and losses in that. So once you've ligated your samples, you do a purification of those smart bells, and you're ready for sequencing. So this is a little bit a look of what you end up with. You've got your lovely sample molecule in the middle that's all free of nicks, and the ends are all clean, and you've adapted on these so-called smart bell adapters. And so this makes the hairpins to make this essentially a topological circular molecule. You bind your or anneal your sequencing primers. They anneal onto these smart bell ends, and then you're going to form a complex with your polymerase at that point as well. And the polymerase has got the streptavidin, which pulls to the biotin that's on the glass of these NMWs, and that's how it pulls everything to the bottom of those little nano holes in the smart cell. And yeah, just to emphasize that when you go into this circular mode, your sequence is essentially both strands of the molecule, because though it's circular, you have got this sort of dumbbell thing. So you're able to sequence, say, the yellow strand, then it goes around the hairpin, and then you sequence the purple strand and back around the hairpin, and then the yellow strand again, and so on and so forth. And this is demonstrated in this diagram on the bottom. The little red dots, imagine these are just random errors, just polymerases make mistakes every now and then, as robust as they are. So they just make random errors in the sequence. Because those random errors are almost never in exactly the same place on the different strands, it just you're able through consensus algorithms to take those errors out, and hence the very, very high accuracy sequencing that you get. There's lots of little animations going on here that show you going through sequencing one strand around the hairpin. And this is a very, very stable form for the polymerase to be in. This is like rolling circle replication in viruses, which is just about the most stable complex that you can form between DNA and a polymerase. So it's very robust. I mentioned if you're wanting to multiplex samples, because you've got a lot of power with the sequencing here on 8 million ZMWs on a smart cell, you're able to multiplex samples if they're small genomes, such as bacterial genomes. So there are barcoded solutions now, so you can multiplex, and there's different ways depending on the applications you're doing of what you can do, you can purchase barcoded the Smart Bell adapters, pre-barcoded sets that you can use, or you can design a sort of two-step PCR method where you you have your lot of specific primers, say, for amplifying a piece of DNA of interest, and you put a tag, a universal tag on that, like an M13 tag, and then have a set of second set of primers to do the second step PCR that binds to the tag and then has your barcodes and adapters on the end. So there's two methods for setting up that multiplexing. Machine itself, it's not a benchtop sequencer. It's a lot bigger than that and a lot heavier than that. You can say it weighs in over 350 kilos, so it needs to be on a floor, and it needs to have very reliable power. So it's linked through a UPS, so that it has very reliable power source. And you know, it's a big sort of cabinet that sits there. And it just needs to be in a space where it's not right up against the wall because of the fans and cooling and things on it. So just a few points to think about where the machine would be placed in your lab. But essentially, once you've got it, the maintenance and things is very low. There's a waste bin for tips and tubes and things that get used on the deck that needs to be emptied. But there's a kind of wizard that goes through when you set up the runs through SmartLink that tells you that your tips are in the right place and your samples and your sequencing plates are all there and the reagents are in date and that the waste bin is emptied and everything's ready to go. So it sort of checks for you, idiot checks for you. So SmartLink, as I said, is the software that's provided with the system, and that really does everything for you from your sample setup right through to the analysis. You do some QCs for your libraries when you clean them up, so you work out how much you've got and you know the average size. So you've run Qubit and a bioanalyzer or TapeStation or something like that, and so you have those pieces of data, and you plug that into the sample setup software, and it calculates the concentrations of things, the amount of sample you need to add, the amount of sequencing primer and the type of sequencing primer that you're going to have. There are options. And the amount of polymerase that you need to bind to make this complex. And then there's a run design part, which is just for setting up the sequencing sample names and that sort of thing within your sample that's going to go on for a particular run for each particular smart cell that you're running. While runs are on, there's a run QC, so you can monitor, and that can be quite useful. And then there's an overall sort of data management, so you can delete old projects or import reference data files, those sorts of things, and obviously a barcode whitelists if you're multiplexing, so it can do analysis for you. And finally, that comes to the smart analysis, and that's the secondary analysis from the sequencing that's coming in. So it will do such things as de novo assemblies for you if that's what you've set it up to do, or it will look for alternative splicing within transcripts and all of those sorts of things. It can do assembly of mitochondrial, of microbial genomes as well, even multiplexed, and form those assemblies for you, those sorts of things. So that all runs through the SmartLink server. You can link multiple SQL systems to that. Not that I think we have any in Australia with multiple systems at the moment, but you can certainly do that. It's a modular system, and you can link your computers obviously to the SmartLink server to do data analysis and you run QC checks and things like that, and monitor runs and analyses and such like. The instrument control software, ICS, as I said, it's like a wizard, really. This is really a screenshot from the little touch screen on the PacBio instrument itself, but it's very similar. You get to your interface on your computer. You get a picture of the deck. It tells you to put tips and tubes and sequencing plates and such like in place. It checks it scans for the barcodes on the plates and things, make sure everything's in the right place, the right way around. You've got enough of everything to run the experiments that you've set up in the run design, and you're good to go. So some of the applications that we have. We will deal with some of these in, as I said, in more detail over the coming series over the next 6 weeks or something, because they're every 2 weeks. So we've got another 3 after today. But we'll skip through and just see some of the things that you can do with a SMART cell. So you can do, as we've said, your whole genome sequencing, and you can do a de novo assemblies and things from that. You can sequence RNA. So there's a kit available to turn your RNA into cDNA. So you do a poly-A capture, make your first strand cDNA. There's a template switch oligo off the end that able to generate your second strand, and then you can sequence that full length cDNA from your sample. Targeted sequencing, so you can make primers, as we mentioned earlier, to amplify specific regions that you might be interested in, to look for SNPs or whatever in that region, or particular pieces of DNA that are very, very hard to sequence because of their sequence repeats or whatever, and you can amplify those and sequence those as a targeted sequencing project, an amplicon type sequencing project. You can sequence from complex populations. So this is such things as microbiome and those sorts of things. So you can look at environmental samples and assay the microbial components in that environmental sample, or it could be a swab sample, a medical swab or something like that, and see which populations of microorganisms are present. And as we mentioned too, because of the way the sequencing works, you automatically gain epigenetic information as well, and you can screen that too. So you can deliberately look at methylation patterns in your samples. And we'll go through just a few examples. This is just a little bit of a favorite of mine, which is why I put in. There's lots of examples of de novo whole genome sequencing out there with PacBio now. But this is one that PacBio actually did in-house. PacBio, from the name, Pacific Biosciences, they're in California. And they decided to sequence the giant California redwood, a sequoia tree. And it's quite a complex genome. It's hexaploid for a start, and it's 27 gigs, so it's big. So it's a challenge, and they actually ended up doing this very, very quickly, from going out into the forest not far from where their base is and collecting pine needles and doing DNA extraction, and right through to running the sequencing HiFi and doing all the alignments and de novo assembly. Was really, really fast. And there's just a comparison of it being done with the other long read sequencing technology with Oxford Nanopore systems, and it was a sort of straight fight really. And it's interesting data. Both are noteworthy, I think, but from the point of view of PacBio in the green color there, the assembly size of haplotype type resolution because of the diploidy of the plant is quite extraordinary, and the contig is very, very good indeed compared to Oxford Nanopore, much more contiguous type assembly, and overall just a more complete genome, even though the coverage was essentially very, very similar. In fact, the ONT data was supplemented with some short read Illumina data as well. So this was a combined data set of ONT and short reads, which was overwhelmed really by the HiFi sequencing with the Sequel II. And the assembly time, I think, is probably the real highlight here, because of the computing power involved to align the ONT compared with the short reads and do that for the scaffolds and everything. It took months. Because of the CCS type data that's coming in, a lot of that sequencing processing is actually being done in real time, effectively on the machine. So when you actually get these perfect reads and they're long, you're able to actually do the de novo assembly in a matter of days. So it's a very, very powerful technique to use. And I'm a plant biologist by training, and so this was a good example for me to see a very complex plant genome being sequenced. Variant detection, we've mentioned that if you're interested in, say, a human genome and looking for differences between different genomes, Obviously, short read sequencing has a lot of problems in certain areas where there are a lot of repeats, and it's very, very difficult to map those short reads. Compared to other long read sequences, but ONT, its accuracy is nowhere near as high as the HiFi, so even that starts to struggle, whereas the PacBio reads is able to cover those unmapped regions and get a fully phased haplotype information from the sample, so you can call very accurately that variant detection. RNA sequencing, we mentioned, is termed isoseq in PacBio world, because of all the isoforms that are available from your alternative splicing of RNA transcripts. So because you're sequencing single molecule and you're sequencing full-length cDNA, you're able to pull out all those isoforms and map those against references where available, or just look at the differential splicing and see if there's any intron exon skips and all those sorts of things. So that's something that's most certainly possible using PacBio sequencing HiFi reads. Those targeted sequencing I was saying, where you can have your specific primers and amplify a region of interest. This might be a region of interest that's been particularly difficult to sequence due to repeats or homopolymers or whatever. So you can amplify these and specifically sequence and again get fully phased haplotype information from various samples. And that's certainly not always possible with short reads, depending on the length of the variant. Here is a 25 base pair variant example, and that's difficult to do with short reads with the confidence you can do with HiFi reads. So onto the microbiome sort of stuff, the complex populations as it's called here. So yes, you're able to look at say an environmental sample or a medical swab or something like that, and look at the microbial communities, and not only what's there, but their relative abundance to one another. And there's some graphs here showing from set samples what the data was able to find. Some of this is using 16S, but it's also possible to do other things, and you can, in some cases, do whole microbial assemblies in this kind of way. But from the metagenomes from the 16S, because you've got the ability to read very long, you're able to read the entire region, 16S, ITS, and right through and get all the variation that's in that region, and get to species or even isolates, subspecies type level, which isn't, again, always possible with other sequencing techniques. Shorter reads, you're only sequencing certain parts of the 16S, maybe V1, V3, or V3, V4, or a certain region. So you see less of the diversity. You may only get to genus or family type identification levels, but it's much higher when you can sequence everything completely with very high accuracy. And then the epigenetics, as we said, you can map the landscape of your genome, as it says here very nicely and very eloquently, for any base modifications. At the moment, the software is predominantly for the A methylation in that GATC motif in prokaryotes. You can look at other types of methylation, C methylation in eukaryotes. The software is still being developed and is not as robust for various reasons, which is more to do with the different types of methylation that's available in eukaryotes, as opposed to just the single 5-methyl A that's in the prokaryotes. Because there's differences, this affects the time delay, and so there's not a simple algorithm that just looks for that set sort of time delay and can call it. It's a lot more subtle, and but the algorithms are still being worked on to improve that, but it can be done. But PacBio are determined to get it better and better if they can. So they're looking more at the subtleties of the various methylated bases and how they're incorporated. And so that's being done. So coming towards the end now, just to sort of round up what you can do with a smart cell, really, just a table here from PacBio and what you can do with a single smart cell 8M on a PacBio Sequel 2. Well, one smart cell's enough for a de novo assembly of a genome up to, say, a 2 gig genome, human genome. It's good to go. Also, a microbial de novo assemblies where you can multiplex up to about 48 microbial isolates on a single smart cell. For variant detection, that's depending on the genome and the complexity and what you're looking for. It's recommended that's probably a project that you run over 2 smart cells rather than 1. But again, it gives you an idea. And I note there is a sort of idea of pricing in this final column that is in US dollars, and exactly how up to date this is probably a year or 2 old, but it gives you an idea. Structural variant detection rather than the full variance detection, so the larger variants for structural calls. Again, a couple of samples up to 2 or 3 gig type genomes can fit onto a smart cell. For the transcriptomics, for the so-called ISO-seq, again, alternative splicing, mapping on a single smart cell, and genome annotation back against the transcriptome databases of up to 8 different samples on a smart cell. Amplicon sequencing will depend a lot on the size of your amplicon and quite what you're doing, but you're able to heavily multiplex different amplicons together, up to 1,000 samples, and that makes the price per sample for your sequencing very, very cheap, just a few dollars a sample. There's also a no-amp kit available from PacBio, so if these regions are really, really hard to even PCR amplify, you can go in with a no-amp sequencing approach and run that on a single smart cell. And again, for the complex populations, for 16S, again, you can heavily multiplex up to 96 samples. Everything's in place from PacBio to do that with the barcoded adapters. And there's other applications as well as I mentioned, the shotgun assembly of metagenomes, and again, with such good accuracy, you get near complete assemblies on a single smart cell. So that's just about the end for me today. I hope you had a good lunch and a cuppa. I'm gasping for one now. But just note, there's lots and lots of information out there now. There's lots of publications using PacBio sequencing. The PacBio website itself's got lots of literature on there and links to lots of publications and things, posters. There's people blogging out there. There's all the user guides and protocols out there for different applications, and even some videos of TED Talks and things where people talk about it and those sorts of things. But do feel free to reach out if you need anything. Reach out to myself or anyone at Millennium Science, and we can certainly try and help you. And just lastly, for anybody attempting this work, there is a nice little site here that's gathered a lot of information from out there in science world, looking at different ways of extracting high quality DNA from all sorts of samples, from plants and animals, microbes, all sorts of things, and the best way to get high molecular weight DNA for your PacBio sequencing. So it's worth checking out extractdna for PacBio.com if you're interested. So that's all for today. I'll hand you back to Sarah, perhaps, to say cheerio.
Thank you. Well, before we say cheerio, Paul, first of all, thank you everyone for joining us. We hope we found this very useful. We've got a few questions. I'm going to start off with a question from Jay. So far, which methylated base that PacBio can detect at the moment?
Okay, so I think I mentioned that at the moment, the algorithms are very much set up for that amethylation in bacterial genomes, so that GATC motif, because there is just that 5-methyl-A that's available or a normal. So it's the algorithms is just a sort of on or off. It's a switch, and the algorithms work very efficiently at that. But other methylation patterns are possible to detect. It's just that the algorithms aren't quite as advanced yet to pick those subtleties between C-methylation types, because there's 4-methyl-C and 5-methyl-C and different things, and they all have slightly different incorporation speeds. So the algorithms are being worked on. At the moment, just A other things are coming.
Okay, great. And we have a question from Paula, who's still actually online. So, do we need to let the sequencing service know we are also looking at methylation? Data comes in BAM format. How can we access the signal information for methylation that was in the HDF5 files for RS?
That's specific, but it's actually a very good question and a very reasonable question too. Yes, I would say in hindsight, tell your sequencing service provider that that you're after that epigenetic information, and that would be good ahead of time. However, most of these providers do keep their data files, and this data is effectively hidden in the metadata because it is that time incorporation. So if you contact them, unless it was a long time ago, I would imagine they still have those files archived, and they should be able to pull that data out, because yes, they've just given you the mapped read BAM files, so they haven't given you that metadata at the back. So speak to them. Hopefully they can help you.
There is hope, Paula. There is hope. Okay. Paula also says, with this high accuracy, do we no longer need Illumina sequencing for polishing genome assemblies?
Oh, you never say no. I don't like the idea of any technology becoming totally redundant. There's a time and a place for everything. I mean, who would have thought with all the next gen, you know, everybody was saying, oh, Sanger's just going to be blown away. Nobody's going to do Sanger sequencing anymore. People are doing it all the time, probably more so now. There's never a lost technology, if you like. So you certainly don't have to use it, but certainly there's a lot of publications out there finding that combinations of different sequencing systems being used together probably gives you the best and perhaps most accurate, fastest and best value for money overall sequencing. But certainly the long read sequencing technology is so powerful now that it does give incredibly good accurate sequencing, and it makes fantastic reference genomes in species where you perhaps don't have a reference. But I wouldn't, even though we don't distribute Illumina, I certainly wouldn't say that there's no need for the technology anymore. That's not on. But yeah, good question though.
Okay. So Paula has another one. Should we be looking for adapters in the sequences?
So I think if you mean from the smart bell adapters where it reads through there, the software is automatically set up to look for those. So it's looking, even from continuous long read sequencing, it's looking to see if it got through the molecule to get round an adapter and sequence back the other way, because it can make some better calls and better judgments there. So the software is looking for those adapters, and it automatically trims, and then it can determine whether it's got multiple passes around the same molecule or it's only really done one very long read and hasn't gone back round and round. But the software is there to look for those adapters and do any trimming and therefore make the consensus reads with really high Q scores.
Okay, great. Hope that answered all of your questions there, Paula. And she says thanks. So we have another one here from Anne-Marie. She says, can we go back and reanalyze our movies for methylation?
So I think that relates back to the service provider question, in so much that if you have those files, yes, you can go back and have a look at those, because the metadata is there for the base incorporation time, and you can trawl through that data to pull up information if you're interested in methylation. Yeah.
Sounds like you might have opened up a can of worms for these service providers. If you cop a few strongly worded emails, you know why.
Well, I'm sure it's somewhere in the contracts that they say we'll store it for however long. If it's still in time, they should still have it, shouldn't they? So yeah.
Okay, there's no more questions here, so we'll leave it there. But if any of the attendees have any additional questions, please feel free to reach out to myself or Paul, and we'll get you an answer as best we can. And we will see you for the next Lunch and Learn. Now, because you've registered for this Lunch and Learn, you're automatically subscribed into the next event. So you will just receive a reminder email 1 hour before, which is in 2 weeks' time. Is that right, Paul?
That's right. I'll try and have a reliable power source at my house by then, so we won't lose power halfway through. My apologies again, everyone, for that.
Not a problem. And thank you very much, Paul. And thank you for everyone for attending. And we'll see you in 2 weeks' time. Bye.
Cheers.